A paper came out this week in Cell Genomics. Three researchers from Oberlin College, MIT, and the University of Wisconsin are claiming they've identified a way to study evolution from before the ancestor of all living things. Not before your ancestors, before the ancestor. The one organism that every bacterium, every fungus, every plant, every animal, every single living thing on this planet traces back to. They're saying some of the genes inside your body right now are older than that. Let that land for a second. You carry molecular artifacts from before the common ancestor of all life on Earth. Not metaphorically, physically, in every cell. So what did these researchers actually find, and why are some scientists calling these genes the only evidence we will ever have of a lost evolutionary world? Let's start with what we know. Every living organism on Earth, from the bacteria in your gut to the whales in the ocean to the mold on your bread, descends from a single ancestor. Scientists call it LUCA, the Last Universal Common Ancestor. And LUCA is not what most people imagine. It's not some simple blob floating in a warm pond. A major study in 2024 dated LUCA to roughly 4.2 billion years ago. That's only about 200 million years after Earth's surface became habitable. And that study estimated LUCA had around 2,600 genes, a full DNA genome, ribosomes, cell membranes, an immune system to fight off viruses. LUCA was about as complex as a modern bacterium 4.2 billion years ago. Now that raises an obvious question. If the oldest ancestor we can trace was already that sophisticated, what came before it? How did we get from raw chemistry to a fully functioning cell in what is, geologically speaking, almost no time at all? That's the gap. That's the mystery. And for decades, the scientists have basically said, we can't look past LUCA. It's a wall. Everything before it is lost to time. This new paper says, not everything. The key is something called universal paralogs. And I know that sounds like jargon, but the concept is surprisingly intuitive once you hear it. So let me walk you through it. You know how you have genes, right? Sections of your DNA that encode proteins. Sometimes a gene gets duplicated, a copying error during cell division, and suddenly an organism has two copies of the same gene. Over millions of years, those copies drift apart. They mutate independently. They take on slightly different jobs. These duplicated genes within the same organism are called paralogs. You actually have a perfect example in your own body. You carry 8 different hemoglobin genes. All 8 descended from a single ancestral gene about 800 million years ago. That one gene got duplicated, then those copies got duplicated, and now you've got 8 versions, all slightly different, all related. Now, most gene duplications are relatively recent in evolutionary terms. They happened within a specific lineage, mammals, vertebrates, whatever. But some duplications are so ancient that both copies exist in virtually every living thing on the planet. Bacteria have them, archaea have them, you have them. If both copies are that widespread, the duplication must have happened before LUCA, before the ancestor of all life. Both copies were already sitting in LUCA's genome when it gave rise to everything else. These are universal paralogs, gene families that duplicated before the last universal common ancestor even existed. And the reason they matter so much comes down to a technical trick that's actually kind of elegant. Normally, when you build a family tree, you need an outgroup, something outside the family to tell you which branch is oldest. If you're comparing humans and chimps, you use gorillas as the outgroup. Simple. But for the tree of all life, there is no outgroup. Everything alive is inside the tree. There's nothing outside it to anchor the root. Universal paralogs solve this, because each copy of the duplicated gene traces its own version of the tree of life. One paralog gives you one tree, the other paralog gives you another tree, and connecting those two trees is a single branch that reaches back to the original duplication event, an event that happened before LUCA. Each copy essentially serves as an outgroup for the other. This was figured out independently by 2 scientists, Peter Gagarin and Naoki Iwabe, back in 1989. It's the only widely accepted method for rooting the entire tree of life. But what Goldman, Fournier, and Cassar are arguing in this new paper goes further. They're saying that branch, the one connecting the two paralog trees, the pre-LUCA branch, carries actual information about what was happening before the ancestor of all life existed. It's a data recorder from a vanished world. And when they surveyed every known universal paralog, they found something striking. Every single one falls into just two categories. Either it's involved in building proteins, the translation machinery that reads genetic code and assembles amino acids, or it's involved in transporting molecules across cell membranes. That's it. Two functions. Out of all the thousands of things a cell can do, the only activities that left detectable traces from before LUCA are making proteins and moving stuff across membranes. Think about what that implies. Those two capabilities, the ability to read genetic instructions and build from them, and the ability to maintain a boundary between inside and outside, were apparently the first stable features of cellular life, the earliest infrastructure that evolution locked in and never let go of. The genetic translation system, in particular, these researchers argue is likely the most ancient molecular system retained in any living thing today. Now you might be thinking, okay, but how many of these universal paralogs actually exist? And this is where the story gets both more interesting and more honest. The answer is not many. Only a handful of universal paralog families have been confidently identified. That might sound like a weakness, but the paper argues the scarcity isn't because LUCA's genome lacked duplicated genes. It's because 4 billion years of evolution has been systematically destroying the evidence. Genes get lost. Sequences mutate beyond recognition. Horizontal gene transfer, where organisms swap genes with unrelated species, scrambles the family trees. Most traces of pre-LUCA duplications have simply been erased, which means the ones we do have are, as one of the authors put it, precious. Gregory Fournier from MIT said it bluntly, "The history of these universal paralogs is the only information we will ever have about these earliest cellular lineages, and so we need to carefully extract as much knowledge as we can from them. The only information we will ever have." That's a heavy statement from an MIT scientist. He's not being dramatic. He's being precise. There are no fossils from 4 billion years ago. The geological record doesn't go back that far. The only evidence that survives from before LUCA is encoded in the genes of living organisms. In your genes. Right now. So what have they actually learned from reading these molecular fossils? One of the most fascinating findings comes from a group of enzymes called aminoacyl tRNA synthetases. These are the molecules responsible for matching the right amino acid to the right piece of genetic code during protein construction. They're absolutely essential. Without them, the genetic code is meaningless. Just letters with no translation. Researchers reconstructed the pre-LUCA ancestor of the enzymes that handle 3 specific amino acids, leucine, isoleucine, and valine. And what they found is that this ancient ancestor couldn't tell them apart. It was, to use the technical term, promiscuous, sloppy. It would grab any of the 3 and just use it. Close enough, which is wild to think about. Modern cells are precision machines. Every amino acid goes exactly where it's supposed to. But before LUCA, life apparently didn't work that way. The machinery was flexible, approximate, good enough to function, but nowhere near accurate. Life didn't start with precision engineering. It started with tolerance, with "that'll do," and somehow that was enough. There's another finding that's even stranger. Researchers looked at the pre-LUCA ancestor of the enzyme that handles tryptophan, one of the 20 amino acids in the standard genetic code. The ancestral enzyme contained no tryptophan at all, 0. The enzyme that would eventually become responsible for incorporating tryptophan into proteins was itself built without any tryptophan, which means tryptophan was a late addition to the genetic code. It wasn't part of the original set. The code we think of as universal and fixed was actually assembled piece by piece over time through a process of trial and addition. The genetic code itself evolved, and Goldman's own lab at Oberlin did something that sounds like science fiction. They took the computational reconstruction of a pre-LUCA protein, one involved in embedding other proteins into cell membranes, and they physically built it. They synthesized a protein from before the ancestor of all life, and it worked. It was simpler than its modern descendants, but it could still bind to cell membranes and interact with today's protein synthesis machinery. A molecule engineered by evolution more than 4 billion years ago, resurrected in a lab in Ohio, and it still functions with modern cellular equipment. The compatibility is remarkable. It tells you something about how deeply conserved these systems are. Now, I want to be careful here because there's a version of this story that goes too far, and I don't want to tell that version. The headlines you've probably seen say, genes older than life itself. That's catchy, but it's not accurate. A gene duplication requires a genetic system to already exist. You can't duplicate a gene if genes haven't been invented yet. What these paralogs are older than is LUCA, the last universal common ancestor, not the origin of life. LUCA is not the beginning of life. LUCA is the oldest point we can reach by comparing living organisms. There was life before LUCA, possibly a lot of life. LUCA is just the bottleneck that all surviving lineages pass through, and this is actually one of the most mind-bending parts. LUCA wasn't alone. The 2024 study that dated LUCA to 4.2 billion years ago also concluded that LUCA lived within an established ecological system. There were other complex organisms around, neighbors, competitors, maybe even predators. Their lineages just didn't make it. They went extinct, and their descendants vanished. Or did they? The new paper raises a possibility that's straight out of a detective story. Some genes in modern archaea show patterns that are consistent with horizontal gene transfer from lineages that no longer exist. Ghost lineages, organisms that went extinct billions of years ago, but managed to pass some of their genetic material sideways into surviving lineages before they disappeared. If that's true, then scattered through the genomes of living organisms are fragments of DNA from evolutionary branches that have been dead for 4 billion years. Messages in bottles from species that lost the survival lottery. The paper doesn't present this as proven. It presents it as a real interpretive possibility based on certain tree topologies. But even as a possibility, it changes how you think about genomes. Your DNA isn't just a record of your ancestors. It might contain borrowed fragments from lineages that have no living descendants at all. Now, I should flag some important limitations because this wouldn't be good science communication without them. First, this paper is a perspective piece. It's a review and synthesis. It's not presenting new experimental data or announcing the discovery of new universal paralog families. Its contribution is pulling together scattered findings and making the case that this should be a research priority. That's valuable, but it's different from a breakthrough discovery. Second, the analytical challenges are real. We're talking about sequences that have been diverging for more than 4 billion years. Over that timescale, the same sites in a gene mutate over and over, eventually erasing the original signal. It's like trying to read a manuscript that's been overwritten dozens of times. There's a technical problem called long branch attraction, where rapidly evolving sequences can look similar by sheer coincidence, tricking the algorithms into drawing the wrong tree. Some critics, including the researchers Forterre and Philippe, have argued that key universal paralog datasets are saturated with mutations, meaning so much has changed that reliable reconstruction might not be possible. Third, horizontal gene transfer is a double-edged sword. It's not just something that might preserve ghost lineage signals. It's also something that can scramble phylogenetic trees, making gene histories diverge wildly from species histories. Even Gogarten, who pioneered the reciprocal rooting method with universal paralogs, acknowledged that the original metaphor of a tree no longer fits the data. Early evolution might have looked less like a tree and more like a tangled web. And fourth, there's a deeper conceptual issue. Carl Woese, one of the most important microbiologists of the 20th century, the person who discovered the archaea, argued that the earliest phase of life might have been qualitatively different from anything that came after. He envisioned a communal stage where horizontal gene transfer was so rampant that individual lineages didn't really exist yet, a kind of collective genetic commons where the concept of a species didn't apply. If that's true, then trying to draw clean evolutionary trees through that period might be forcing a modern framework onto something that didn't work that way. These are real concerns. They don't invalidate the approach, but they define how much confidence we should place in any specific claim about pre-LUCA biology. The signal is faint, the noise is enormous, and the tools, while improving, are working at the absolute edge of what molecular phylogenetics can do. That said, the tools are getting better. And this is where the paper gets genuinely exciting about the future. AlphaFold and similar AI systems can now predict the three-dimensional structure of reconstructed ancient proteins. That's a game changer because structure tells you about function in ways that sequence alone can't. You can reconstruct an ancestral protein, predict how it folds, and start asking, what could this thing actually do? Deep learning might also help identify new universal paralog families that have diverged so far they're invisible to traditional search methods. Combine that with the ancestral protein resurrection work, actually synthesizing these ancient molecules and testing them in the lab, and you're looking at a field that's moving from pure speculation to experimental science. Betul Kachar, the third author, has literally inserted a 700-million-year-old protein into living bacteria and watched them adapt to it. Now imagine pushing that approach even further back. Before LUCA, before the tree of life had a trunk. There's also an implication here that the paper doesn't spend much time on, but that's hard to ignore. All three authors are deeply connected to NASA's astrobiology program. Kachar leads a NASA astrobiology center. Fournier has published on detecting biosignatures on Saturn's moon Enceladus. Goldman is affiliated with the Blue Marble Space Institute of Science, and the timeline they're describing is suggestive. Earth became habitable around 4.3 to 4.4 billion years ago. LUCA was already a complex cell by 4.2 billion years ago. That's a window of maybe 1 to 200 million years from habitable planet to something as sophisticated as a modern bacterium, complete with an immune system. If life can get that complex that fast on Earth, it becomes much harder to argue it would be a unique fluke. A one-time accident. The rapid emergence of biological complexity looks less like a miracle and more like something chemistry just does when the conditions are right. That doesn't prove life exists elsewhere, but it shifts the probability in an interesting direction. So where does all of this leave us? We have a paper that is, in its own author's words, a call to action. Not a single dramatic revelation, but a case that these handful of ancient, duplicated gene families deserve far more attention than they've gotten. Because they might be, and this is the part that sticks with me, the only window we will ever have into a chapter of evolution that no fossil, no rock, no geological record preserves. Somewhere around 4 billion years ago, before the ancestor of every living thing existed, genes were already duplicating, proteins were already working, membranes were already forming boundaries between self and everything else, and the descendants of those ancient molecular machines are still running, inside bacteria, inside archaea, inside you, doing roughly the same jobs they were doing before the tree of life even had roots. We talk a lot about looking for signs of life on other planets. Turns out some of the oldest signs of life anywhere are sitting inside your own cells. We just didn't know how to read them until now. If you want more of the newest science that's rewriting what we know about where we come from and what's out there, there's always another discovery waiting.